The expression of a recombinant protein in bacteria requires to insert the gene of this protein into our bacterial cells so that they produce the corresponding protein as if it was one of their own. To do so, the gene of interest inserted in an expression vector is transferred into bacterial cells by a process known as transformation. The transformed bacterial cells are then grown to a critical mass, and when enough cells are present, the expression of the protein of interest is induced. In this lesson, I will introduce you to this process step by step to go from the expression vector carrying our gene of interest to the bacterial cell pellet that contain our recombinantly expressed protein. When bacterial cells have to express a foreign protein, this may be stressful or even toxic for them. To avoid stressing them, which may impair their proper growth, one uses an induction system so that the protein of interest is only expressed when a sufficient amount of cells has been grown in unstressful conditions. The most classical system used for protein expression in E. coli is the T7 promoter IPTG system. In this system, the coding DNA sequence of our protein of interest is inserted by cloning in an expression vector, like the PET vector, so that it is under the control of the T7 promoter. This means that it will transcribe into mRNA by the T7 RNA polymerase. This polymerase is a bacteriophage polymerase. It is not present in bacteria. But the bacteria we use for recombinant protein expression have been genetically engineered so that they possess the gene for the T7 RNA polymerase in the genome. However, this gene is not constitutively expressed as it is inserted in the lac operon. So under normal conditions, the gene for the T7 RNA polymerase will thus be repressed. It will only be transcribed upon addition of lactose or analogues in the culture medium. In our case, we will use isopropyl beta-D thiogalactoside, IPTG in short. When a sufficient mass of bacterial cells has been grown, we will add IPTG in the culture medium. This will induce the transcription and the production of the T7 RNA polymerase. That in turn will transcribe the gene of our target protein and allow for its production in large amounts since the T7 promoter is a strong promoter. Now let's move on to the practical part. It's very important to work under sterile conditions all throughout the process to avoid contaminating our bacterial cells with surrounding bacteria. And for this purpose, we use a laminal flow hood that is decontaminated with bactericidal solution and ethanol before use. To avoid stressing the bacterial cells and diminish the efficiency of the transformation, it's also important to maintain the cells on ice, as well as the reagents that will be all in direct contact with them, such as the solution of the expression vector carrying our gene of interest. First, an aliquot of competent cells is retrieved from storage at -80 degrees Celsius and is thawed on ice for 5 minutes under the hood. If transformation is performed by electroporation, an electroporation cuvette is also cooled down on ice. Then 100 to 300 nanograms of the plasmidic DNA is added to the cells, and the DNA is left to mix with the cells for a few minutes on ice. For heat shock transformation, the mixture is incubated for exactly 30 seconds at 42 degrees Celsius on a heating block, and then immediately put back on ice. For electroporation, the mixture is transferred into the pre-cooled electroporation cuvette. The cuvette is wiped off with paper and placed in the electroporator. A voltage of 1.8 kilovolts is applied for a few seconds. These two procedures allow the expression vector to enter the bacterial cells. And then around 300 microliters of bacterial culture medium, LB or equivalent, is added to the transformed cells. The mix is then transferred into a new clean and sterile Eppendorf tube, and the cells are incubated at 37 degrees Celsius for an hour under vigorous shaking. The expression vector inserted in the bacterial cells generally carries a resistance cassette that confers resistance to the transformed cells to a particular antibiotic, in our case, kanamycin. This incubation period of 1 hour without antibiotics is called outgrowth. It allows for cell recovery and expression of antibiotic resistance. After incubation, the mix is recovered and the cells are spread on an LB agar plate containing the appropriate antibiotics. Depending on the amount of DNA used for the transformation, only part of the transformation mix may be spread in order to obtain single and well-isolated colonies. The plate is then incubated overnight at 37 degrees Celsius in a static incubator. It is best to use dry and warm plates. The next day, we can prepare a culture medium for large-scale production of bacteria. We prepare large flasks with 2.5 liters of sterile medium. We usually prefer to use 2xYT medium as compared to standard luria broth. This medium is richer and allows to grow more cells. In the afternoon, we recover the plate with the transformed bacterial cells. As you can see, single colonies are visible. We can now make an overnight start culture that will serve as a seed for our large-scale cultures tomorrow. Under the flow hood, we prepare a flask with 50 ml of 2xYT medium supplemented with the appropriate antibiotics. Then we pick up a few colonies from the plate and inoculate them into the flask. It's important to select a few as they may not all express the protein of interest to the same level. Then we pick up a few colonies from the plate and inoculate them into the flask. It's important to select a few as they may not all express the protein of interest in the same level. The flask is then incubated overnight at 37 degrees Celsius in a shaking incubator so that the cells grow to saturation. Our star culture has grown to saturation and we can now inoculate a small volume of this into the big flasks of culture medium, supplemented with the appropriate antibiotics. We usually make an 150-fold dilution of the start culture. The flasks are then incubated at 37 degrees Celsius under shaking until bacteria reach their exponential phase of growth. Exponential phase of growth is reached when the optical density of the bacterial culture at 600 nanometers is around 0.5 to 0.7. After a few hours of incubation at 37 degrees Celsius, we regularly monitor the optical density with the help of a spectrophotometer. For this, 1 ml of the bacterial culture is inserted into a spectrophotometer cuvette under sterile conditions. A cuvette containing the 2xYT medium without bacteria is also prepared and is used to make the blank reference. The optical density of our culture is then measured. If the value is within the expected range, cells can then be induced for protein expression. Otherwise, they are put back at 37 degrees Celsius to grow until the desired OD value is reached. Once bacterial cultures have reached the exponential phase of growth, expression of our protein of interest can be induced by adding IPTG in the culture medium. As explained previously, IPTG will bind to the LAG-I repressor and remove repression on the T7 RNA polymerase gene. This polymerase will be produced and will in turn allow transcription and production of our protein. Once IPTG has been added, the cultures are incubated overnight at 18 degrees Celsius under shaking. The temperature is lower to avoid stressing unnecessarily the cells. A sufficient mass of cells has been grown now, and the cells can only focus on producing our protein of interest. The next day, the cell cultures are recovered and centrifuged to obtain dry pellets that will be stored at -20 degrees Celsius until purification. At this stage, we don't need to work under sterile conditions anymore. We use large-scale centrifugation with 1-liter pots. Once the cells are all pelleted, we transfer the pellets in 50 ml tubes, 1 to 2-liter culture pellets per tube, and we store them at -20 degrees Celsius.